content of these seeds. Beta-conglutine is the major storage protein in these seeds. And you know that as the seeds germinate, it undergoes, all storage molecules undergo a catabolic process by which they are cleaved into smaller molecules. In the case of proteins, and you can see here in an SDS page gel, that we begin with beta-conglutine with bands that have a high molecular weight, and during germination, these larger polypeptides will be cleaved into smaller polypeptides, and then they disappear because they will be turned out into amino acids, which will be used by the plant to grow. In the case of lupinus, it has the unique characteristic that it was found among legumes. It's unique. It has an intermediate accumulation of a polypeptide that you can see here, that it does not exist in the seeds, it does not exist in an adult plant, it only exists during a short period of time. And that polypeptide is really the molecule that has the fungicidal activity, and we call blood. That's why the name ProBlood for the product. So we spent a long time studying the mode of action of this active ingredient. We work along with several research institutes throughout the world, with several PhD students and master theses as well, and we have published all these scientific results in several scientific publications in peer-reviewed international journals. So you have here the references if you want to read the science behind the knowledge that I'm going to explain, which is the mode of action of our active ingredient. This is a very rough scheme. I made it myself, so it's not very nice. But it's the simplest way to explain the mode of action of blood. You can see here the fungal cell with the cell wall, cell membrane, the space in between where you have the glycosylated residues from the membrane proteins, and here you have the cytoplasm. So blood is able to cross the cell wall. We don't know exactly how, but we know that it disturbs the cell wall, although not in a lethal way. Once it reaches this space, it has an affinity for the glycosylated residues because it has a lectin activity, and it is able to cross as well the cell membrane. We also don't know how, but we know that it disturbs in a very hard way the cell membrane. So then we have blood in the center, in the cytoplasm of the cell, and already the cell is debilitated with damages at the cell wall and at the cell membrane, but not lethal. Once inside the cell, there occurs the major mode of action of this molecule. I just want to remind you that inside the cell, we have a lot of enzymes that are responsible for the metabolism of the cells. And these enzymes require cofactors to be active. And most enzymes use as cofactors divalent cations like calcium, copper, iron, magnesium, manganese, and zinc. So only when they are activated by these cofactors, they can act and make the reactions that are responsible for cell metabolism. What blood does is that it has a scavenging activity like a chelator, and so it binds these divalent cations, and these cations are no longer available for the enzymes. And therefore, many reactions in the cell are blocked because there are no more divalent cations available, and therefore the cell metabolism is blocked, and the cell ends by dying in the end. So this is a completely novel mode of action, and it is also a multi-target mode of action because it acts not only at the cell wall and at the cell membrane, but most importantly, inside the cells, it acts upon so many different reactions that the cells are they are not able to find a pathway to overcome this inactivation. So this is a very robust mode of action against the development of resistance in the cells. This new mechanism of action was already acknowledged by the FRAC committee as being completely novel and multi-target, and in 2016 they created a special category, a new category just specifically for blood. And presently we are in BM01, which is the biologicals with multiple modes of actions, and therefore our product can be mixed with all other registered products because it has a completely new mode of action, and it can be used in IPM programs as well. Of course, that we have also made all the required studies in terms of toxicity, toxicological and ecotoxicologically. In terms of human exposure, toxicology studies were made, both in vitro and in vivo, and the toxicological profile was proven to be compatible for human manipulation, ingestion, inhalation, and so on. We also did a lot of ecotoxicological studies as required to evaluate the environmental safety of the product, and it was found to be safe for the environment. Most importantly, our product is very readily biodegradable because it is a natural molecule. All the extract is natural, and the main route of biological degradation is by the proteases that are present in the soil, and the end products, of course, are safe molecules for the soil as well. Just to summarize the most relevant properties of the product, we have this ProBlood is exempt from maximum residue levels, which allows to reduce the use of conventional products and therefore minimizes residues. It is environmental friendly, as I said, and it has an absence or a very short pre-harvest interval and re-entry interval. That will depend on the country, so it's 0 or 1 day. Although it is a contact fungicide, it has translaminar activity, which is quite important, and it is tank mix compatible with most fungicides, insecticides, and herbicides, and fertilizers. It has a preventative period of up to 8 days and a curative period of also up to 8 days, but of course at low levels of disease pressure, because it should be used mostly as preventative. It is not, it does not perform miracles in the field, so if you have a high infestation, you don't need a fungicide, you need a miracle. It is very stable at high and low temperatures, and it's not sensitive to UV. As I said, it has a novel and unique mode of action, so it can rotate with all other frag codes, with a multi-side effect on fungal cells, prevents the development of resistance, and so it can be used as a new and effective resistance management tool. It has a broad spectrum, Maria will talk about that, and it's active against most life stages of diseases like spores, Epha, mycelium, and Austoria, and it has a fast knockdown effect of less than 24 hours. And now Maria will talk about the efficacy.
1 minuto e mezzo.
Sì. Buongiorno a tutti, sono Maria Dimartino. Molto velocemente, perché mi interessava più che altro farvi vedere quella che era la modalità d'azione nuova di questo prodotto. Molto velocemente passiamo a quelle che sono le principali prove di efficacia che sono state condotte in Europa per valutare la selettività e l'efficacia del ProBlood. In particolare vi riporto quelle che sono le principali, più significative prove condotte su vite, fragole e pomodoro per il controllo di Botrytis cinerea, su drupace per il controllo di monilia. Si riportano quelli che sono i rilievi effettuati, il numero di prove che sono state prese in considerazione e quella che è l'efficacia media percentuale di ProBlood a confronto con gli standard chimici di riferimento che sono stati utilizzati nelle prove di efficacia. Come potete vedere dai risultati si evidenzia un ottimo controllo di ProBlood nel controllo delle infezioni causate da Botrytis cinerea e da monilia, con dei valori di efficacia che vanno dal 60% fino quasi all'80% nel caso della Botrytis su pomodoro, e con dei valori che sono paragonabili a quelli degli standard chimici di riferimento utilizzati nelle prove. Altre prove sono state sempre condotte su vite e pomodoro per il controllo di infezioni da malbianco. In questo caso vedete che in presenza di percentuali medie di infezioni su testimoni non trattati da medio-alte, vedete dei valori di efficacia che
altri 30 secondi.
Sì, perfetto, che si attestano intorno al 69% fino a quasi l'88% di efficacia, e anche qui dei valori di efficacia paragonabili agli standard chimici di riferimento. Altro target molto importante è il brusone del riso. Anche qui vedete l'ottimo controllo del prodotto, con valori di efficacia con rilievi effettuati su foglie e su pannocchia paragonabile comunque non significativamente diversi dagli standard chimici di riferimento, quindi con un'ottima attività nel controllo di infezioni causate da Piricularия oryzae. Quindi questo è per riassumere un po' quelle che sono le caratteristiche del prodotto. Vi sottolineo principalmente l'ottima selettività che è stata riscontrata sulle colture, non è stato rilevato nessun sintomo di fitotossicità, l'ottima miscibilità del prodotto con i principali prodotti fitosanitari e fertilizzanti fogliari che sono presenti in commercio. Dalle prove di trasformazione e quindi da quelle che sono le prove di vinificazione è emerso anche nessuna interferenza negativa del prodotto sui principali processi di mostificazione e vinificazione e per i processi di trasformazione che portano appunto alla produzione di succhi di frutta, purè e marmellata. Questa sarà la prima proposta di etichetta con le dosi indicate, le colture e le avversità. Ovviamente è in corso un'ulteriore attività di ricerca sul prodotto per prossime estensioni di etichetta. Grazie per l'attenzione.